Hi, my name is Brian Luna. I'm here today at the University of California, Irvine from the James Lab. Today I'll be talking to you about double-stranded RNA injections into adult Aedes aegypti female mosquitoes. So the procedure consists of using a manual setup and injecting the adult females into their thorax. Double-stranded RNA injections can be used to knock down numerous genes. In our lab specifically, we're using it to knock down genes related to steroid biosynthesis. Now I'm going to be talking to you about the preparation of the needles. Now I prepare the needles using these borosilicate glass capillary tubes. Now these capillaries are pulled on our Sutter microinjection puller. And the next step after that the needles have been pulled is to calibrate them using water. The next step in this process is the calibration of the needles. So once the needles have been pulled from the Sutter puller, they come out looking like so. The final product will have the needles clearly marked with a black Sharpie marker. Now in order to standardize the markings, each needle is loaded with a set amount of volume by a micropipetter. Calibration of the needles is important, or else we will not know the exact concentration or volume that we are injecting into each mosquito. For the purpose of this demonstration, I've loaded the capillary tubes with a food coloring dye instead of the typical double-stranded RNA suspension. The double-stranded RNA can be loaded into the needles by inserting the needles into the solution, and capillary action will draw out the fluid. Now I'm going to be talking to you about our injection setup. It's composed of a 50 milliliter syringe, a stopcock assembly, attached to a rubber hose, and a glass capillary adapter. The stopcock assembly can be adjusted to 3 separate positions. This first position is the resting position. The second position is in